this turned up loud enough? I'm not used to the lapel mic. Can everyone hear me in the back? Okay, great. So I guess I'm going to be the guinea pig for this new policy, so we'll see how this goes. Okay, so to begin my presentation, my dissertation work, I'm just going to give a brief outline of what to expect from this talk. So I'm going to start with the theoretical framework about life history evolution and life history trade-offs. Then talk about my model system that I use, the Tephritid fruit flies, and then go into the actual results that I found in using the female fruit flies and the males. So the kind of the foundation of any life history is the timing at which an organism is born and then dies. So during this period of time, we have stage-specific rates of maturation, growth, and death, especially in holometabolous insects. With the maggots will pupate into pupae, and then you'll have a period of eclosion, and then the adults will eclose and then mature into reproductive maturity. These life history traits are under strong selection by the environment, as well as influence on the phenotypic expression. So that life history traits are extremely plastic, especially in insects which are ectothermic. So the environment really regulates the metabolism. So of course you're expecting to see some sort of effect of the environment on the lifespan and then these kind of timing of these life events. Also, natural selection is expected to act on life history traits and drive the evolution of adaptive trade-offs and other traits in the organism. In addition to the environment affecting the life histories of organisms, there's internal constraints that are inherent to every organism. One of these constraints is that there's a basis or a bias based on the evolutionary history of that organism. So it's constrained by its ancestry. So if it comes from a line of short-lived organisms, it's not automatically going to be able to live 100 years, for instance. Aside from these phylogenetic constraints, there's individual constraints that they experience. This is through trade-offs. A trade-off is when there's a benefit to one trait, so they maybe allocate some fitness benefit with reproduction, but then at the same time there's a cost and a loss of fitness in the other traits. This is often due to energy limitations in the ecosystem. This idea has actually led to a lot of hypotheses to describe and make predictions about trade-offs in ecosystems, and it led to the development of this kind of optimal resource allocation model. Since all physiological functions have to draw from the same resource pool, those resources, nutritional resources that are present in the environment, and it has to use those and then allocate those to whichever function will best optimize its fitness. Now this is a very generalized model. Of course there's going to be more physiological functions such as homeostasis and other things, but here I'm just going to, again, generalize it across reproduction and survival, which represents somatic maintenance. It's expected, and is also seen with a lot of experimental evidence, that as food resources decline in the environment or they become less quality, the probability of survival increases, and at the same time, reproductive rate declines or halts altogether. This is thought to be an adaptive life history trade-off because it allows the organism to avoid the cost of reproduction if that reproductive ability isn't guaranteed. That cost of reproduction is as current reproductive effort increases, there's a decrease in the probability of survival. And by decreasing survival, you have a shortened lifespan. And by shortening the lifespan, there's fewer opportunities to mate in the future. So again, by avoiding those poor conditions, maybe at some point in the future they'll improve, and then you can begin your reproductive effort. Unfortunately, you just can't shift this indefinitely because there's a second link between longevity and reproductive ability, and that is through reproductive senescence. As the individuals grow older, the reproductive machinery kind of breaks down, and you see a loss of age-specific reproduction. So there's this two-fold trade-off occurring where you don't want to waste survival budget on reproduction that's not guaranteed, But you don't want to delay your reproductive ability too long because you'll lose a greater proportion of fitness or lose the ability to mate altogether if you die. So to test these kind of ideas, I worked in the model system of the Tephritid fruit flies. Now, Tephrididae shouldn't be confused with the Drosophilids. Those are actually a common group of organisms that, like Drosophila madagascar, is included and is used as a model system in many genetic and other fields. These fruit flies are phylogenetically distinct, and they've been separated by a great period of evolutionary time. There's about 4,500 species of Tephritid, and they're spread across 500 genera. The 3 species I used make up 3 of these genera. So we have representation for the genus Ceratodus, Bactrocera, and Anastropha. These 3 species are of grave economic importance, especially here in California, as these are continually invading the state, and there's evidence that these are potentially established. So using these 3 species allowed me to kind of work with several different Tephritid experts in the world, and the selection of each of these species was based on the location I was conducting the research and also the specific questions I was interested in asking. So to begin the actual results section, I will talk about these life history trade-offs in the female Tephridids. So in female insects, there's a cost of reproduction. As I outlined before, that cost is a reduction in lifespan. This has been linked to not only copulation, where the insertion of the male genitalia can damage the fly and then reduce the survival probability, but also to the production of eggs, where each egg consists of some sort of energy budget that detracts from the survival, thereby reducing the overall survival. So because of these 2 different costs, a great thing for the Tephridids, which makes them a useful model organism for testing these trade-offs, is that copulation isn't needed to maximize reproductive ability. So it's not required that a male inseminate the female to allow her to produce the total suite of eggs she would anyway. So this allows for the disaggregation of the cost of copulation and the cost of eggs. And this is what I did in my study. All these females that I applied to these treatments were maintained as virgins, so I can just get a sense of how the eggs affect the trade-offs. So I initiated trade-offs through simulating poor reproductive conditions in 2 qualitatively different mechanisms. The first mechanism is through denying the host. And what this does is it doesn't allow the organism to oviposit the eggs. It does allow for some amount to be matured in the ovaries, but at some point the number of eggs that can be retained is limited, and then oostatic hormone is produced to prevent the further maturation of eggs. The second way in which reproduction can be delayed is through the denial of the dietary precursors of vitalogenesis. So this actually prevents the ovaries from being able to mature, and no eggs can be actually generated until that protein source is given. And this is expected to have very different effects on life history trade-offs, because there's also potentially a detrimental effect on the physiology and the somatic maintenance of the individual flies based on these poor conditions. So to test these effects of these trade-offs, I used 2 species, the Mediterranean fruit fly and the melon fly. The females were chosen for these 2 species because of their variation in their life history traits. The med fly is a relatively short-lived species, while the melon fly has a much longer lifespan. Also, there's interesting differences in their reproductive histories, which I will show here. So in this graph, we have the average eggs laid per female across the age of the females. So what we see here is these are results from the control, the reproductive controls from these experiments. So that when the med fly's life's reproductive ability is characterized by this really quick period of maturation, reproduction is maximized early in the lifespan, and then the rest of the lifespan is characterized by reproductive senescence. The melon fly, on the other hand, it takes a bit longer to reach maturity, but it doesn't have this dramatic peak that the med fly does, and that the rate in which reproduction declines with age is diminished compared to the med fly. So any differences in how these 2 species react to life history trade-offs which show how these different life histories influence the ability to make decisions about somatic maintenance and reproductive ability. If there's any similarities, it might show that there's some conserved responses to these poor conditions within these two genera. To figure out what the cost of egg laying was, I began one treatment where flies always had access to this host, which is a papaya piece, which is a common host to both species and is very available in Hawaii where this research was conducted. So this control, these females received the papaya daily until death. To see how limiting the oviposition rates affected lifespan, I constructed a treatment structure where there's two different deprivation periods of host. The first deprivation period occurred for a 7-day period. So at eclosion, the flies did not receive the host, which is represented here by the lack of the red ring around the circle. At age 7 days, the host is introduced and then provided daily until death. A second deprivation period was a 14-day deprivation, where the same pattern occurs, and at age 14 days, the flies are provided with the papaya pieces. To really determine what the cost of egg laying was, there needed to be a second control, and these were flies that never had access to the host plant. So this would be some baseline survival where there's no cost. When you actually look at the life expectancy of the medfly under all these treatments, there's a very surprising result. There's actually no difference between the individuals that are never laying eggs and those that are continuously laying eggs. So this shows there's a lack of egg laying costs and a lack of cost of reproduction. And this is really what not expected. And there's a few explanations for this that I've kind of mused over. And one may be that these flies are already so short-lived that the cost of reproduction is really minimalized, and we just can't see it across these treatments, and maybe more individuals in the treatments would help drive the significance. Additionally, there is a potential that the cost of reproduction is being incurred by these flies, and that they're maturing the eggs, and by retaining them, there's the same amount of lifespan reduction as if she was continuously laying the eggs. Now when you look at the life expectancy of the melon fly, we see a different result. Here is what we were expecting to see with the med fly, where you actually have the longest lifespan, life expectancy associated with those individuals that were never laying eggs, and then the shortest life expectancy with the continual reproduction. Unfortunately, these deprivation periods don't improve the lifespan, so there's no trade-off going on. These flies are allocating the resources provided to them to produce eggs in the ovary and then retain them until reproduction can occur. So when you look at how these affect the age-specific reproduction rates, we see kind of two common trends. Again, here we have the eggs laid per female with age in days. This gray line here in the background that's shadowed, that represents the reproductive control that I showed earlier. The overlaid bars here represent the reproductive ability of the treatments. So when host was delayed for 7 days, we see that the flies are losing this period where the reproduction was peaked, and that there's some level of retention, which is demonstrated here by this spike in reproductive ability. Even though there's this huge spike here, there's no ability to kind of compensate for this missed ability early. And then we see the same thing. So this delay during the first 2 weeks then misses the entire period where reproduction is maximized. And then there is a slight peak of reproduction, which is actually lower than what's seen in the 7-day. But they are never able to kind of allocate resources in a way that prevents the total loss of life reproductive ability. In the melon fly, we see a slightly different result in the 7-day deprivation period, where the reproductive rates are almost identical in the treatments and the control. And that's because the melon fly didn't reach reproductive maturity until later in the deprivation period, so it didn't have time to develop those eggs and then retain them. So that's why we don't see the peak that's associated with the deprivation periods in the other two treatments. Now we do see that for the 14-day delay, and it follows really the same pattern seen in the med fly, where you miss the period in which reproduction would be maximized, there's a slight spike, and then reproductive rates kind of mirror that of the controls. Now to see how that second mechanism influences survival and life expectancy, I had a second control where flies were given this full diet, which is comprised of a yeast-derived protein and a carbohydrate from sugar. And this provides all the nutritional requirements to maximize the survival of these flies. And so the control involved giving the flies this full diet every day from eclosion until death. Now, when we delayed the full diet for 7 days, we started the flies on a sugar-only diet, represented here by this empty circle. Following a 7-day deprivation period, that full diet was provided until death. And we did the same with the 14-day dietary deprivation period, where at 14 days we give the full diet until death. And then we had a negative control, in which sugar diet was provided every day. And this would be able to see how these dietary deprivation treatments affect the lifespan relative to individuals that always had full diet and then those that never did. So with the medfly, we see that any access to that sugar diet is very detrimental to the life expectancy. However, if the full diet is encountered by 7 days, there is a slight reversal in that loss of life expectancy. And so it kind of provides a rescue effect. However, if the delay period is longer than the 7 days, there is no longer any benefit to having that protein's access. So you can see the 14-day deprivation period has the same life expectancy as the flies that never receive protein. Now, when we look at the effect of these deprivation cycles on the melon fly, we see a similar result where that sugar diet is extremely detrimental if it's encountered early in the lifespan, but the melon fly has no ability to kind of correct for that, even if protein's provided within 1 week of eclosion. So the melon fly is very dependent on that protein source early in life to really maximize its lifespan. Now, when we look at the effect of delaying vitellogenesis on the age-specific reproduction, we kind of see a different result from when host axis was just deprived. Now, again, we have the eggs laid per female with age, which is the x-axis, and then the control is the shaded area with the treatments overlaid on top of that. When the protein was delayed for a 7-day period, the medfly loses reproductive ability at both ends of the lifespan. So at the beginning of life, there's no ability to produce eggs, obviously, and then they have a shortened life expectancy. So it really smashes that period of reproductive ability that the medfly has. What's interesting is as soon as the protein is provided, the maturation occurs, and then you kind of get this period where they have similar rates of reproduction as the control, but then it drops quickly. With the 14-day deprivation period, we again see that you can't have any reproduction until the protein's provided, and then you lose lifespan at the tail end of the reproductive curve. What's interesting here is that when the delay is for 14 days, it actually shifts this whole reproductive schedule over relative to the 7-day period. So it's actually everything shifted 7 days over. So in some way, these medfly are able to kind of allocate resources away from current reproduction in favor of late life reproduction, and then diminish that effect by shifting the reproductive ability over. The melon fly shows similar results, except that we don't have that loss of life expectancy at the end because they are longer lived. And again, the 14-day deprivation period causes the reproduction to take longer period to reach maturation. When you actually look at the rates of senescence, the medfly under these 2 deprivation periods show a reduced rate of decline with age to reproduction compared to the control. So there's another level of the trade-off where they're able to slow the rate at which reproduction will be lost. But again, that's not enough to overcome the initial detriment of the sugar diet, but it will allow for some fitness to occur in that shortened lifespan. So in summary of the reproduction in females, we see that the cost of egg production isn't as simple as a 1-to-1 trade-off between 1 unit of energy for survival and 1 unit of reproductive energy. And there's complex trade-offs occurring where that reproduction cost can be avoided in the medfly specifically. There's also this clear importance of timing, both if the protein availability and also the host access. And so what this would be implied is that in that first 2 weeks that these flies eclose, and this is applicable in the wild, is they're going to be really active to try to find their dietary precursors and their host access. Otherwise, they're going to have greatly reduced fecundity. And then finally, there's this interesting result that there's a trade-off that allows for senescence to be slightly slowed, but is only present in the medfly. So once I kind of saw these results in the females, I was wondering what had been done in looking at life history trade-offs in males. And just my surprise is there's very little looking at any types of trade-offs involving reproduction and survival. I think this is related to kind of a dogma that's associated with the disposable soma hypothesis, where males are often kind of assumed just to be sperm donors flying around and just trying to maximize their reproductive ability. But current research is showing that that's probably not the case for every species. And males do show cost of reproduction that's more than just related to competition for mates and then competition for resources. And these costs of reproduction are associated with behaviors such as pre-mating behaviors, as well as the physiological processes of reproduction, such as sperm production and pheromone production to actually attract mates. So because there's this cost of reproduction, it kind of seems that it'd be obvious that there'd be trade-offs between survival and reproductive ability if the male doesn't think it can maximize the reproductive success at a given time. So how I thought about driving these reproductive trade-offs is through limiting the mate access, so not giving them females, and also giving them access to these poor diets. Mate access alone shouldn't drive trade-offs because the males will still initiate calling behaviors to try to attract mates that could potentially be in the environment. But the diet type was expected to perhaps have the same effect on the females, where some precursor would no longer be available to the reproduction and then kind of shift the reproductive ability. So to test this first concept of male life history trade-offs, I used the male Mexican fruit fly. This fruit fly is reared in Tapachula, Mexico for sterile release and control of this fly throughout Central America and whenever it occurs in the United States. So I had access to plenty of flies for experimental units. So what I first did was give them access to that poor diet and see how that drove their life expectancies. So here we have the life expectancy on the y-axis and the two diet types that they were given. The yeast plus sugar is the same full diet that the females received in the prior study, and it's known to really maximize the male's reproductive ability, its calling rates, and its survival in prior experiments. And then I gave them this restricted diet in which they only received the carbohydrate from sugar. And what's seen is there's actually a 2-week loss of life expectancy associated with these poor dietary conditions, so that there is going to be potential for some trade-off. If they're losing life expectancy here because of the diet, maybe they won't try to allocate resources to reproduction so they can survive until the conditions improve. So when I looked at the costs of reproduction in the Mex fly, when they were provided that full diet, we see that there is strong evidence that the act of copulating and reproducing with a female is detrimental to life expectancy. And so when males were virgins, they lived almost 3 weeks longer than males that had continuous access to virgin females. So if this trend is carried on into the sugar diet, it's expected that the sugar-fed mated flies are going to have very short life expectancies. Unfortunately, we don't see that. There's actually no difference between the males that are kept as virgins and those that have continuous access to mates. Now, there's two potential explanations, one being biologically interesting and the one being not. The uninteresting thing is they're just not mating. So of course they're not going to be acquiring a cost. The female's there, they don't bother it. But we're interested in the trade-offs here. And so if there is some reproduction going on, there's evidence then that there's some trade-off between the survival and the reproductive ability, and in some way they're avoiding that cost of reproduction. And so I actually did try to quantify the male reproductive effort. And how I did this is I had to maintain a pool of virgin females that were aged to 7 to 14 days, which is kind of the prime receptiveness to male copulation. But they're also not going to die on you when you're monitoring for egg laying. And I would give each male in the reproductive treatments one of these females, and he would have access to her for 24 hours. Following the 24-hour period, that female was then removed and placed in a separate holding cage with an oviposition site, and she was monitored for 15 days. All the eggs that she did lay in that period were collected, and those were then monitored for a further 10 days. How I quantified reproduction then is if the eggs hatched, it was assumed that that male successfully inseminated the female at the day 30 or 25 days earlier when she actually had the access to the male. While no hatch would be the opposite. There was a day of no reproductive success. What we find when we compare the effects of the diet on the rate of reproduction, the rate of reproduction, by the way, is the proportion of males to actually inseminate that female. So we weren't able to actually quantify it because you would have to count the larvae produced by the female, but then you would have a bias of the female's reproductive ability kind of distorting the male's reproductive success. So it was more of a qualitative, a yes or no response. So on average, the full diet of fed flies were more likely to inseminate a female at any age. And then the sugar diet, there was a slight reduction. But this reduction isn't substantially less than that of the full diet. And this is not evidence that they're not mating. So they're clearly reproducing with these females, and they're somehow, in some other way, avoiding the cost of reproduction. A second interesting finding that came out of the looking at the age-specific insemination success is we have actually outlined the entire reproductive ability of a male for most of the lifespan. I had to end it at 7 days because I had to come back to the United States from Mexico. So again, we have the insemination success, the proportion of the males at each age that successfully inseminate a female. And what we see is that across the two diet types, the protein, which is the dark square, and the empty triangle, is that there's the same pattern regardless of that diet. And that's this period where reproduction is maturing early in the lifespan, and then it reaches some peak early, about midlife, and then following about 35 days, there's evidence of reproductive senescence in the males. And this finding is pretty novel. There's not a lot of work done in looking at lifetime reproductive success of males. I could only find one other paper where they actually tried to figure out if males experience senescence. And I think that this result here really should help us rethink the concepts of disposable soma, because if males are indeed senescing during their lifespan, that's going to influence their reproductive effort for the rest of the life. And so, in summary for this portion, the protein is very important to maximizing lifespan, but it's not needed for reproduction. And then there's this clear evidence for reproductive senescence, and so that's really kind of an exciting result, and hopefully this drives more research into kind of figuring out what males are doing with reproductive effort throughout the entire lifespan. So since the disposable soma theory or hypotheses aren't supported by the male Tephridids, I was interested in seeing if a related hypothesis would be supported in another Tephritid species, and this is the terminal investment hypothesis. What the terminal investment hypothesis suggests is that the probability of survival is directly linked to reproductive effort. So as the risk of death increases, and therefore the probability of survival decreases, the organism is expected to ramp up its reproductive effort. And this is also kind of falling in line with that disposable soma hypothesis. Because reproductive effort is then increasing, I expect that there's going to be this feedback loop where then the cost of reproduction is magnified, survival decreases further, and then reproduction is further increased. And so I set out to test this by increasing the mortality artificially. And since just the effect of age didn't drive an increase in reproductive ability, I decided to see if an artificial risk of mortality would have effects on reproductive behavior as suggested by the terminal investment hypothesis, and how that would affect that cost of reproduction. To do this, I used the Mediterranean fruit fly. I only studied this in males. The med fly was really useful for this because of their clear way of demonstrating reproductive effort. And I'll get to that a little bit in just a little bit. So how I caused this artificial risk of mortality was through actually amputating a portion of the front leg of these flies. So following carbon dioxide-induced anesthesia, I removed the tarsomere segments 2 through 5, and that included the claw on the end of the leg there. And this was expected to cause a reduction in lifespan, but the amount of damage was hoped to be lessened or low enough that it didn't affect the reproductive ability too much. So what we find is that yes, the amputation does increase the risk of death and thereby reduces the life expectancy. So here we have the males that are solitary, so those are our control group, just to determine what amputation does. And amputation reduces the life expectancy by just over 2 weeks. Now when combined with female access, we see again a cost of female access between the intact individuals, but then we don't see significant interaction of the terms, where instead of this being dramatically reduced through some synergistic effect or a linear effect, the loss of life expectancy of the amputated pair males is exactly the same as that, or not exactly, but it's very similar to that of the intact individuals. But it is less than as if amputation occurred without female access. So this was interesting to figure out what's driving this difference. Why is each reproductive event not driving this further, or is there an increase in reproductive effort as suggested by that terminal investment hypothesis? And to do this, I did behavioral assays where I was looking at male reproductive effort through measuring pheromone signaling, and this is what the males are really useful in doing, is that reproductive effort can be quantified by counting how often a male will signal. This might be a little low for everyone to see, but what the male will do is it will flex its abdomen, and in doing that, it extrudes this little pheromone bubble. And in doing that, it attracts females. So the more often it does that, the more reproductive effort is demonstrated. So what I did is I kept these males with only males. There was no copulation going on. And then I would report the proportion of male signaling every hour, beginning at 8 a.m. and ending at 3 p.m., which is kind of their period of activity. To see if there was an effect of mating, I did the same thing where I compared the mating success of intact and impaired individuals by housing these males in cages with 2-to-1 male to female ratio, or female to male ratio, so there was no competition for the mates. So anyone that wanted to mate would be able to get access to the female. And what I did is every hour I would observe the proportion of those males that were successfully mating. What we see is the terminal investment hypothesis is not supported. So here on the y-axis is the proportion of individuals calling at any given hour, and that is recorded for the first 5 days of life, beginning at age 2, because at age 1 they're not mature yet and not calling. So there is a strong age effect where reproductive ability and effort increases as the males age, but there's no effect of amputating them. So if the terminal investment hypothesis was supported, we'd see some increased level of effort then to kind of counter that increased mortality risk. Also, it's surprising that it didn't cause a detriment, so that the impairment really doesn't reduce the willingness of the male to try to find a female, and this just shows how they prioritize reproduction, even if they're limping around and they're still trying to get that fitness assured. Now, when you actually look at the copulation rates, so here instead of proportion calling, we have the proportion of those males copulating with age, we see now that the amputation does affect the ability to mate. With the age effect still intact, the impaired individuals with that amputation have reduced success at all these ages of observation. And this is, again, against that terminal investment hypothesis, but there's a different biological explanation which is equally interesting. So when you look at the actual age and hourly specific copulation rates, so again, here's the proportion of those males copulating over time, beginning at 8 a.m. and ending at 3 p.m., we see that the overall pattern remains intact, so that the the damage doesn't influence any of the circadian patterns, but it reduces the copulation success at every age and every time. And through observing these flies, it shows that, or I've seen that it's the females that are really driving this reduction in copulation ability. The females have a lot of choice in the medfly, so that the calling to attract the female first brings her in to kind of investigate the potential suitor. But once she sees that he's kind of this deformed individual missing his leg, the female will either turn away uninterested, or if the male does try to mount, she'll actually kick him out. It's kind of a funny behavior to watch, and the male kind of is dejected and walks away. And so that female choice is really important, and if you're damaged, these males aren't increasing the reproductive effort, and they're also losing this ability to attract the mates. So in closing of this section, again, the cost of impairment is that reduction of lifespan, but it also reduces the fitness of the organism. So it's in the organism's best interest to avoid kind of being injured in the environment. And being injured has repercussions more than just being easier to catch and eat and having a shorter life, but you're less likely to then be able to maximize your reproductive fitness. The terminal investment hypothesis, again, is not supported by that lack of an increase of reproductive effort. And so that's those general findings for that study. Now, when we look at the kind of the overall themes seen through these different species and these different experiments, we see that the cost of reproduction is more complicated than just allocating one energy unit away from survival towards reproduction. And there's all these different levels of, there's all these intricacies in how trade-offs influence the reproductive ability and how survival is often not prioritized compared to the reproductive success. The reproductive senescence, we see interesting results in how the female flies are able to slow the rate of reproductive senescence, and that we actually have reproductive senescence in males. And what this does is it provides more information on making predictions about how life history traits will evolve in environments, and really kind of show that we shouldn't rely on some of these older hypotheses relating to resource allocation, especially when there's assumptions that reproductive effort will increase indefinitely until death. And then, again, we have to really rethink the disposable soma hypothesis and the related hypotheses in that, because as organisms age, there is that breakdown of reproductive ability. So hopefully this information can be used to and be applied to looking at how life history traits evolve in natural environments. This is work that I have started, but unfortunately I didn't have time to complete. And there is evidence of environments really influencing how trade-offs occur, with more variable environments selecting for life history trade-offs and resistance to poor conditions. So with that, I'd like to thank you all for your attendance, and specifically my major professor, James Carey, for all of his support and guidance over the last 4 years in helping me kind of construct these interesting questions and ways of testing them, as well as my thesis dissertation committee, which include Frank Zalem here in the department, and Roger Vargas at the USDA Pacific Basin Agriculture Research Center in Hilo, Hawaii. I also like to thank my collaborators here at UC Davis, the lab technicians that helped me in my experiments at the Hilo Research Laboratories, the research leader at the Ecosur, Mexico station where I conducted the Mexican fruit fly work, and then all the lab technicians there and at the rearing facility. All my research was funded by the National Institute of Health under the National Institute of Aging, by the biodemographic determinants of lifespan PO1 program. Thank you again for your attendance. I think I have time for questions.
Okay, we got 5 minutes.
Yes.
So if you did these same tests with a species that has a much more constricted host range, would you expect to have the same temporal patterns of trade-offs?
So,
so for instance, if you look at cherry fruit fly,
Yeah.
there's not cherries around that long. Are they going
I would
to be better or
imagine,
worse?
okay, I would imagine that they have more flexibility. So the medfly, as you kind of suggest in your question, it has a huge host range. So I imagine if there's not one host available, it will go seek out another. And they will actually even deposit into really horrible hosts that limit their lifespan and make their larvae really small and kind of wimpy. Something more specialized, there may be more selective pressure to shift that reproductive ability because they're really dependent on the availability of that host. So if they're temporarily off from their host, they're going to need to extend the lifespan and that reproductive ability. So if there's selective pressure driving that, I would imagine you would see a greater ability to extend reproductive lifespan.
So you didn't, you talked about the disposable soma theory, but I'm not sure that you actually ever defined it.
Oh, great.
Or maybe I missed that.
No, I guess I know.
And then a second follow-up question, related question is, so what can I tell the National Institute on Aging? What have you learned about aging?
Okay. So the disposable soma theory was proposed back in the 1970s by a researcher at Kirkwood. Basically what his hypothesis was is that based on that limited amount of resources in any ecosystem, there's going to be some way of prioritizing the resources that maximize the fitness. And what's expected for males is because of, or what's expected for males is that as they increase in age, the reproductive ability will also increase, just kind of either linearly or logarithmically or whatever the kind of risk of death is. And so the females have been shown to not follow that pattern, and that's been kind of long established. And criticism of that theory, now I kind of have further evidence that the males don't follow that either. Now what I learned, what to tell NIH, so that the patterns of kind of just expression of lifespan are more complicated than just some genetically driven trait. There's a lot of phenotypic plasticity that occurs during that lifespan, and that then kind of drives how the rate of senescence occurs, and also can factor into the evolution of further life history traits. So that if an individual is more plastic, it's going to have an adaptive benefit if the conditions are negative than an individual that's more constrict in its life history traits.
What else did you learn about amputations? And I'll just make a comment here because I give talks on this. You know, the impairment is a big deal in aging research. And, you know, we'd have talks here where you could use pesticides, say you killed 10,000 insects, and of course they're writhing in pain with it. You get up and say, I snipped the tips off of 10 fruit flies. Everybody goes, horror. So I've given a talk like that. Anyway.
Okay. An interesting response to the amputation is it's not, the loss of life expectancy is not due to the shock associated with losing a leg. So actually the survival probability is about the same as the intact individuals for the first 2 weeks of life. So it's not an acute health response, it's actually a chronic kind of accumulated response over time. So in some way the accumulation of disability days, that's what drives that loss of life expectancy. But the resistance to that damage is actually shown to be very dependent on age. So that the young individuals show this chronic effect, where I had also aged individuals that I didn't quite have time to include that information in the presentation. But the older flies, they respond the exact opposite. As soon as they lose that limb, survival just kind of plummets. So they lose that ability to be resistant after they've been aged a significant amount.
Anything else? Anyway, I thought it was a great talk, so thanks for coming and thanks for
kind of work with several different Tephritid experts in the world, and the selection of each of these species was based on the location I was conducting the research and also the specific questions I was interested in asking. So to begin the actual results section, I will talk about these life history trade-offs in the female Tephridids. So in female insects, there's a cost of reproduction. As I outlined before, that cost is a reduction in lifespan. This has been linked to not only copulation, where the insertion of the male genitalia can damage the fly and then reduce the survival probability, but also to the production of eggs, where each egg consists of some sort of energy budget that detracts from the survival, thereby reducing the overall survival. So because of these two different costs, a great thing for the Tephridids, which makes them a useful model organism for testing these trade-offs, is that copulation isn't needed to maximize reproductive ability. So it's not required that a male inseminate the female to allow her to produce the total suite of eggs she would anyway. So this allows for the disaggregation of the cost of copulation and the cost of eggs. And this is what I did in my study. All these females that I applied to these treatments were maintained as virgins, so I can just get a sense of how the eggs affect the trade-offs. Sort of effect of the environment on the lifespan and then these kind of timing of these life events. Also, natural selection is expected to act on life history traits and drive the evolution of adaptive trade-offs and other traits in the organism. In addition to the environment affecting the life histories of organisms, there's internal constraints that are inherent to every organism. One of these constraints is that there's a basis or a bias based on the evolutionary history of that organism. So it's constrained by its ancestry. So if it comes from a line of short-lived organisms, it's not automatically going to be able to live 100 years, for instance. Aside from these phylogenetic constraints, there's individual constraints that they experience. This is through trade-offs. A trade-off is when there's a benefit to one trait, so they maybe allocate some fitness benefit with reproduction, but then at the same time there's a cost and a loss of fitness in the other traits. This is often due to energy limitations in the ecosystem. This idea has actually led to a lot of hypotheses to describe and make predictions about trade-offs in ecosystems, and it led to the development of this kind of optimal resource allocation model. Since all physiological functions have to draw from the same resource pool, those resources, nutritional resources that are present in the environment, and it has to use those and then allocate those to whichever function will best optimize its fitness. Now this is a very generalized model. Of course there's going to be more physiological functions such as homeostasis and other things, but here I'm just going to, again, generalize it across reproduction and survival, which represents somatic maintenance. It's expected, and is also seen with a lot of experimental evidence, that as food resources decline in the environment or they become less quality, the probability of survival increases, and at the same time, reproductive rate declines or halts altogether. This is thought to be an adaptive life history trade-off because it allows the organism to avoid the cost of reproduction if that reproductive ability isn't guaranteed. That cost of reproduction is as current reproductive effort increases, there's a decrease in the probability of survival. And by decreasing survival, you have a shortened lifespan. And by shortening the lifespan, there's fewer opportunities to mate in the future. So again, by avoiding those poor conditions, maybe at some point in the future they'll improve, and then you can begin your reproductive effort. Unfortunately, you just can't shift this indefinitely because there's a second link between longevity and reproductive ability, and that is through reproductive. Is this turned up loud enough? I'm not used to lapel mic. Can everyone hear me in the back? Okay, great. So I guess I'm going to be the guinea pig for this new policy, so we'll see how this goes. Okay, so to begin my presentation, my dissertation work, I'm just going to give a brief outline of what to expect from this talk. So I'm going to start with the theoretical framework about life history evolution and life history trade-offs. Then talk about my model system that I use, the Tephritid fruit flies, and then go into the actual results that I found in using the female fruit flies and the males. So the kind of the foundation of any life history is the timing at which an organism is born and then dies. So during this period of time, we have stage-specific rates of maturation, growth, and death, especially in holometabolous insects, with the maggots will pupate into pupae, and then you have a period of eclosion, and then the adults will eclose and then mature into reproductive maturity. These life history traits are under strong selection by the environment, as well as influence on the phenotypic expression. So that life history traits are extremely plastic, especially in insects which are ectothermic. So the environment really regulates the metabolism. So of course you're expecting to see some sort of senescence. As the individuals grow older, the reproductive machinery kind of breaks down, and you see a loss of age-specific reproduction. So there's this two-fold trade-off occurring where you don't want to waste survival budget on reproduction that's not guaranteed, but you don't want to delay your reproductive ability too long because you'll lose a greater proportion of fitness or lose the ability to mate altogether if you die. So to test these kind of ideas, I worked in the model system of the Tephritid fruit flies. Now, Tephritid shouldn't be confused with the Drosophilids. Those are actually a common group of organisms that, like Drosophila madagascar, is included and is used as a model system in many genetic and other fields. These fruit flies are phylogenetically distinct, and they've been separated by a great period of evolutionary time. There's about 4,500 species of Tephritid, and they're spread across 500 genera. The three species I used make up 3 of these genera. So we have representation for the genus Ceratodus, Bactrocera, and Anastropha. These 3 species are of grave economic importance, especially here in California, as these are continually invading the state, and there's evidence that these are potentially established. So using these 3 species allowed me